Hello, everyone, and thank you for attending today's webinar. Ethylene Oxide Sterilization Requirements have changed. What you need to know. And this is being brought to you by Medical Design and Outsourcing and Nelson Laboratories, a Sotera Health company. Before we begin, I just want to cover a few housekeeping items. At the bottom of your console are multiple application widgets. If you have any questions during the webcast, you can use the Q&A widget. We will capture all questions, and we will have a Q&A session at the end of the presentation. If we don't get to your question, we will answer it via email as well. And a copy of today's slide deck is available in the resource list widget, which is the green folder at the bottom of your screen. You can expand your slide area by clicking on the maximize icon at the top right, sliding or dragging the bottom right corner as well. And again, an on-demand version of this webcast will be available approximately one day after. It can be accessed using the same audience link that was sent to you earlier. And of course, you can always tweet with us at #MDOWebinar. We welcome all your comments. My name is Heather Thompson. I'm the Senior Editor of Medical Design and Outsourcing. And today I'm speaking with Paul Littley, the Consulting Manager at Nelson Laboratories, and David Gilbert, the EO Sterilization Consulting Study Director at Nelson Labs. Today's webinar, we're going to take a comprehensive look at the single lot release approach, the factors that determine what's necessary, and how requirements in Annex E of the ANSI AAMI ISO 11135 have changed. Our speakers are going to navigate some of the considerations for the execution of a single batch release process, and they'll also assist in development of a testing plan, including sample size requirements for performing a single batch sterilization process. With that, I will turn it over to you, Paul.
Thank you, Heather. Hello, and thank you for joining us today for this webinar on ethylene oxide sterilization, revision of Annex E single batch release. My name is Paul Littley. I'll be co-hosting with David Gilbert, who is also more commonly known as Gib. Today's presentation, we will be discussing Annex E of ISO 11135, which is a normative section of the standard for single lot release. Annex E specifies the requirements for sterilizing and releasing products from a sterilization process without performing a sterilization validation of the process first. The process is considered fully validated and is specific to the lot or batch under test. For today's presentation, we'll start with an introduction of ethylene oxide sterilization, and from there we'll go into a few definitions, EO sterilization validation, the more traditional standard approach for validating a sterilization cycle, and then from there we'll actually jump into single batch procedures, how to conform to the standard, product release conforming with the standard, and then the updates to Annex E of the new revisions. So for the introduction. So ethylene oxide is a gaseous sterilization process. The ethylene oxide molecule is a cyclical epoxide ring, which is easily opened due to the string bond angles. EO is a gas at 51 degrees Fahrenheit or higher in temperature. Very versatile chemical intermediate, and again, very, very reactive. The advantages of ethylene oxide sterilization are that it's low temperature, extremely material friendly. Ethylene oxide does follow the traditional overkill approach as most gaseous sterilization processes do, providing a large safety margin for sterility assurance. Ethylene oxide has excellent penetrating abilities, very small molecule. EO has a very long history of regulatory acceptance. It is considered an established Category A method, spanning back into the 1940s with the history of the sterilization. The revalidation interval is fairly long at a year, as the new expectations, and it's very scalable from small or large volumes and large or small batches. Some of the disadvantages, unfortunately, are EO residuals, I think we're all aware of. EO is also very toxic. It is a carcinogen. It's very flammable, as we spoke about. That epoxide ring is easily open, making it very, very reactive. Unless you have a parametric release, the release interval can add time to your overall sterilization process. And of course, last but not least, the of EO needs, which need to be handled by the contractor. The four main parameters of an ethylene sterilization cycle are temperature, relative humidity, gas concentration, exposure time. For temperatures, we're typically talking about temperatures in the range of 50 to 55 degrees Celsius. So fairly low temperatures with regards to some of the other gaseous products. Relative humidity levels are typically anything above 35% or greater. Your common relative humidity ranges are going to be anywhere from 50 to 80%. Gas concentrations will be anywhere from 400 or 500 all the way up to 700 or 800 milligrams per liter. And exposure times for a typical device, not very difficult to sterilize. You're looking at about 2 to 4 hours of gas exposure, which would give you about a 12-hour chamber time for overall sterilization. Some things to remember about ethylene oxide: it is a gas in, gas out process, so the packaging needs to allow for the air removal, must be permeable, and please note the typo there with permeable. Got caught there. But anyway, the packaging must allow for humidity and sterilant penetration as well as off-gassing. The packaging must be comparable with multiple pressure changes, as ethylene oxide sterilization is typically performed under a vacuum or subatmospheric. Packaging should not increase the bioburden levels, and for most processes, companies like to validate or allow their packaging, validate their packaging to be able to withstand 2 more exposures in the event of sterilization cycle failure. Now we'll go into sterilization definitions and some of the definitions that will be covered and used during the rest of the presentation. The first sterilization definition is a biological indicator. Biological indicator is the meter stick for ethylene oxide sterilization. The definition of a BI is an inoculated carrier that contains a known population and a known resistance to the relevant sterilization process. For ethylene oxide, the indicator organism is Bacillus atrophius. In these 2 slides, you see how Bacillus atrophius is typically grown out on the plate on the left in these large orange cultures, and then on the right, on the right slide, and actually the right test tube, you see the turbidity and also that classic orange pellicle on top of the growth media, both indicating growth of Bacillus atrophius. The BI indicator for EO is inoculated with 1 × 10⁶ colony-forming units per carrier, and again, the indicator organism is Bacillus atrophius. The next definition is process challenge device. Process challenge device is really a surrogate for your product. It simulates the resistance of your product, and the proper definition is that it's an item designed to constitute a defined resistance to a sterilization process and used to assess the performance of the process instead of using a product. Again, it really is a coupon or a surrogate for using a live product, which in some cases can be very costly. Sterility assurance level. Sterility assurance level is the probability of a single viable microorganism occurring on a product or on an item after sterilization. It is a quantitative value, and generally we're talking 10⁻⁶. In some cases, there are allowances for 10⁻³ for some special products, but typically what we are looking for is product. For an SAL of 10⁻⁶, in general terms, it is the probability of the existence of 1 non-sterile unit out of 1 million units sterilized. Again, adding a very high level or high safety and safety margin product. The next definition is decimal value reduction value. And this is the time or dose, in this case for ethylene oxide, is the time required to achieve inactivation of 90% or 1 log of a population of the test microorganism test. In the graph on the right, we can see on the y-axis we have the microbial count or microbial population, and then on the x-axis we have time. As we look at this, we see that in 50 seconds, we're able to achieve 1 log reduction of the population, of the known population. So that's giving us a d-value of 50 seconds. Talk a lot about overkill concept, but the overkill concept or overkill approach, as it's identified in Annex B of the standard, is also known as the half-cycle method. And this is a process which delivers a minimum of 12 spore log reduction to the biological indicator in the full validation or full cycle of the sterilization process. The half cycle is where we actually have achieved full kill of the BI. In this next slide, we see that the graph is depicting basically what your half cycle and your full cycle should achieve. So again, on the y-axis, we have microbial or microbiological population. Right there at the middle, we have 10 to the 0. If we look at the top of the graph on the y-axis, we see that that's 10 to the 6. So that's representing our microbial population of the BI. Start to move across to the right, we look at exposure time in hours. At the 2-hour point, we see that the biological indicator line intersects with the 10 to the 0 of the population, and that's indicating in half cycle that you've achieved complete kill of your BI. If all parameters and process parameters and variables are held consistent, it can be assumed that in the second half of the cycle, making it the full cycle, that second 2 hours, you would have an additional 6 log kill, making that an overall kill of 10 to the 12th, or spore log reduction, a 12 spore log reduction. Section 3 is EO sterilization validation. This is what a typical validation is going to look like, a standard validation. Again, the reason why we bring this into light is because in the second half of this presentation, Gib will be covering the process for a single lot batch and what that looks like and how it fits into and adheres to the requirements of achieving sterile cycle or showing that your product is sterile after the single lot batch release. In a typical or traditional validation of a sterilization process, we refer to that as a performance qualification. We're not going to be talking about installation qualification or operational qualification for the purpose of this discussion today, but we will talk about just the PQ and what that looks like. Typically speaking, a standard validation will consist of 1 sublethal cycle, 3 half cycles, and 1 full cycle. And in the graph below, we have basically the pressure versus time, showing the overall pressure and interaction of time versus pressure in a sterilization cycle. You can see that we pull down from atmospheric down to a vacuum, evacuating all the air. And as we spoke about earlier, EO being highly reactive and highly flammable, this is why we operate at subatmospheric pressures. But if we want to really look at this closer and what the sublethal versus half versus full cycles look like, we can look at the center horizontal line. That represents gas dwell. In a sublethal cycle, it is a shortened cycle where sublethal means that we are actually expecting fractional or partial growth of our biological indicators or our process challenge device combination. In the half cycle, a little bit longer of a cycle, or half the full cycle, we're expecting full BI kill. Then, of course, the full cycle is representative of the entire dwell time there in this graph. These last spikes in sawtooth waves are the gas washes to flush out the EO and reduce those EO residuals after the gas dwell phase. And again, in the overkill process, just as a reminder, in the half cycle, we are reducing that BI to basically a 0 organism count, and then doubling that time for the full cycle. That is what makes the overkill concept. Typical testing during the validation for the performance qualification. Prior to any of the actual cycles that we run, we will look at your product for bioburden testing, and Gib will go a little bit more in depth on that subject once we get into the actual process or procedure for single batch release. But again, bioburden testing is just quantifying the number of organisms on your product. A lot of times we'll speciate as well to see if we can actually get a species of the organism, so we have a more known concept of what organisms are on your product prior to sterilization. During your sublethal cycle, again, BI sterility is performed, but this is also where we will perform product sterility. Product sterility is basically to show that your BI-PCD combination is greater in resistant rather than your product naturally occurring bioburden. From the sublethal cycle, we'll run 3 half cycles, and in all 3 half cycles must show BI sterility. Other tests that are performed during the half cycle will be temperature and humidity distribution studies, and then if you are running a traditional internal process challenge device and external process challenge device, with the external being for product release, then you would establish the relationship between the sublethal cycle and the half cycle as well. The last cycle, or last portion of the PQ, will be your full cycle. Again, typically you'll run at least 1, if not 2, for your product to demonstrate the ability of your product to withstand 2 back-to-back sterilization. Common tests for the full cycle is going to be, again, BI sterility. Residual testing, again, this is where we're going to test for EO residuals on your product, ensuring that your aeration phase is adequate for proper gas. And then we'll also be looking at your final temperature and humidity distribution, at the physical performance, if you will, of the sterilization process. In this next section, single batch release procedure, I'll turn the presentation over to Gib for further.
Thank you, Paul. So for clinical batch release, you must keep in mind that as I go through these slides, I'll reference single lot release as a clinical batch or batch release. Just keep in mind that I do intend those terms to be synonymous. So I apologize if I go away from the single lot release as it's referenced in the standard, but I do mean them to be the same thing. Single lot releases are a good way or means to perform research and development of new product if you need a claim of sterility on those devices. And it's also good to have a claim of sterility on for clinical units that you will be using for clinical trials. So if a product meets these conditions, then it's a good means of sterilizing product and actually having a sterility claim. Must emphasize that single lot release is not a means for routine sterilization and should be only for clinical trials or for the research and development as we. Prior to performing any type of clinical batch release, it is a good idea to to look into the composition and design of your product, as well as the packaging, the bioburden, and the eventual load density that you will be sterilizing either at a test facility or a contract sterilization facility. And you must understand or at least identify any type of sensitivities that your product may have. Sometimes it is a good idea to perform some feasibility testing to understand the functionality of your product to EO sterilization prior to performing clinical batch release. So that way you do have that understanding that your product will be functional. Particularly if it does have a pharmaceutical or some type of drug coating, you want to take that into consideration, as temperature, relative humidity, and the EO gas concentration may have an ill effect on the functionality of the device. So single lot releases, Paul was mentioning earlier, is based on the overkill method. Unlike a validation approach where you will validate a cycle and expose product to a single full cycle process, you will actually, for the single lot release, be exposing the load to both the half and full cycles. The half cycle process should be at least half that of the dwell time of your full cycle. Also, you should take into consideration in reducing parameters for your half cycle so they are worst case when compared to your full cycle parameters. And going into that detail of a cycle, how that is determined, the cycle length, since you don't have any cycle development work, you will need to monitor the sterilization process with either an inoculated product or a process challenge device, abbreviated as PCD. You can choose either method to monitor the sterilization process, but in order to use a PCD, there's some additional upfront testing you need to consider. But there are advantages in using a PCD over inoculated product. When inoculating product, we will seed the product with a BI strip that'll be from a third-party manufacturer. We will either inoculate directly sutures, wires, or the product itself with a third-party manufactured inoculum. We try to use the manufactured biological indicators first, as there are some known inconsistencies when using spore inoculum, and we try to avoid the use of inoculum to avoid those inconsistencies that are known. When we place or add the inoculation locations to your devices, we consider the product's configuration. We consider the lumens, any closures, mated surfaces, and understand that with inoculated product, with there not being any type of cycle development data to support which site may be more difficult to sterilize than another, we must inoculate multiple locations if there are locations that are in question to be a challenge to EO sterilization. So we'll not just choose one potentially, but we may choose multiple locations on the devices. Also, when using inoculated product, we are making an educated guess on the resistance of your product because there is no cycle development. So the cycle parameters that are recommended to use for the sterilization of the device is contingent on the configuration itself. Without any type of cycle development data, there's no means to fine-tune the sterilization parameters. Therefore, you would have a very robust process as you would be conservative in recommending lengthier cycles, higher gas concentrations. And when choosing the inoculation locations, like I said, you need to choose wisely, as the FDA will commonly look and question the rationale of choosing those locations for the single lot release process. The other method of monitoring the sterilization process is via the process challenge devices. These process challenge devices are representative of your product's resistance. In order to develop a process challenge device or a PCD, you must do upfront testing called a a comparative resistance and a bioburden resistance. What these tests do is you again inoculate the product and compare that inoculated product to these surrogate devices, and you choose a PCD that is representative of the product's resistance. Ideally, you want it at least equal in resistance or just a little bit more difficult to sterilize than the most difficult to sterilize location. So some of the advantages of developing a PCD and disadvantages are as follows. When it comes to a single lot release and you do have a process challenge device developed, you will use fewer devices. In the slide here, we see that inoculated product is needed if you are not using a PCD. Usually you'll use at least 8 inoculated devices for every clinical batch release. That quantity is based on load volume, and it increases as the volume increases. Product sterility would be satisfied via the comparative resistance and bioburden resistance, as we would perform product sterility to show that the natural product bioburden is less resistant than the PCDs, than the candidate PCDs. And then BF would also be performed as part of that assessment for PCD. Another benefit is you would develop a more distinguished cycle for the sterilizing your product. You wouldn't have to perform an educated guess or have very robust parameters. You can develop a cycle that is more geared to the resistance of your device itself. So it would have lesser EO exposure, thereby having lesser residuals, lesser aeration, and it helps accelerate the sterilization process. The other benefit, and this may be the best out of all of them, is you can use these data for an eventual validation. As long as your product will maintain its same configuration. There's no manufacturing changes, no material changes. The data that you develop in development of the PCD itself can eventually be used and leveraged for a validation effort. So it would satisfy part of that microbial performance qualification, the MPQ. The disadvantages is it does add a little bit more time on the front end prior to a clinical batch release being performed, and there are additional costs. So with having explained how a cycle is determined and how a product is used for monitoring the sterilization process, we'll now go into sample testing and quantities. As mentioned, inoculated product or PCDs are going to be used. That quantity is based on your overall product load volume. The temperature and relative humidity probes will also be used. Those quantities are based upon load volume as well. So when you are planning for your batch release, you'll want to initially take note of an approximate volume that you'll be sterilizing, so that way you can have an understanding of how many devices you're going to need to compromise for the inoculated product and know how many probes you'll need. Bioburden enumeration, even though it's not a bioburden-based sterilization process, you still need to do bioburden enumeration to confirm that the population of your natural product bioburden is less resistant or less in regards to the biological indicator that's used within the inoculated product or the PCDs. And we need a minimum of 3. Product sterility, we'll need to do that as part of the clinical batch release. And then also we'll need to perform BF if not already performed to support the product sterility results. Bacterial Endotoxin is also a requirement for your device if it has a pyrogenicity claim. Depending upon how the product is going to be used, is going to determine if that BET or bacterial endotoxin testing is going to be required. And then of course you'll always have to perform the residual analysis of your product. And keep in mind that these products are randomly chosen from the load from the actual product lot. So predetermining sample quantities or samples to use for testing are usually frowned upon. So here's an example of the inoculated product or PCD quantity, the table that is outlined in Annex C, table C.3. So in the table, if you look at the right-hand side of the table, you'll see it goes by load volume, and those load volumes show a specific quantity that you'll need to use for your MPQ and routine control if you have a routine control use. Something that you'll want to understand is with every clinical batch release process, we will be seeding fresh inoculated devices or PCDs into the load prior to each half and full cycle. So here's an example for your half cycle process. If your load is 3 cubic meters, we'll need to use a minimum of 9 monitors as outlined in the tables of Annex C. And then with the full cycles, you use half the amount. So if you have a 3 cubic meter load, half of 9 would be rounded up actually to 5 and not 4 monitors. It is recommended to use an excess quantity to cover the loss or the compromising of one of the monitors during the sterilization process. That includes the shipping. Temperature probes are the same aspect as the inoculated product. There's a table in Annex C that provides recommendations, and as well as for the relative humidity probes. These tables are to be used. If you do not use these tables to calculate or estimate how many probes or inoculated products you will need to monitor the sterilization process, then those quantities need to be rationalized. So going into the bioburden enumeration, this is performed prior to any type of sterilization effort. We evaluate the product bioburden for the aerobic bacteria, spore-forming bacteria and fungi at a minimum. You can include anaerobes as part of the bioburden enumeration, but we're not too concerned with anaerobes when it comes to EO sterilization. However, as a conservative effort, you can consider evaluating anaerobic bacteria. Product sterility, you'll need 10 to 20 units for the product sterility testing. Depending upon how you would like to test your product sterility, kind of really drives how you would like to evaluate your bioburden as well. You would either test the 10 units or 20 units, however many you submit for testing, and you test them either per a USP method or per an ISO method. The USP method tests in both soy types, or it tests in two media types, a soy broth media and a thioglycate media. The ISO method tests solely in soy media. So a lot of clients like to correlate the data between bioburden and their product sterility data. So if you do perform USP, you'd want to consider evaluating and having anaerobic bacteria data, since you will have anaerobic media being used as part of the sterility test. And the same goes with the ISO method. Usually you'd exclude your anaerobes as part of your bioburden. BF is done to confirm the results of the product sterility test. You want to ensure that the media volume that is being used for your product sterility is not going to inhibit or cause a false negative. And you would also want to ensure that any product materials don't create or cause a false negative. So that's the essence of the BF test. 3 or 6 samples are used for a BF test. If you choose the USP method for product sterility, you will need to default to having 6 samples provided. If you choose the ISO method, then 3 samples would be needed to perform and corroborate the product sterility test results. A lot of clients choose to perform the ISO method, as this uses fewer samples and it is cheaper. Bacterial endotoxin is only needed if you have a pyrogenicity claim on your device. With a bacterial endotoxin claim, this is performed after the full cycle process, after both half and full cycle processes. So we want to ensure that the sterilization process does not impact those levels, and that is why it's performed post-sterilization. We need 3% of the batch release lot, and with a minimum of 3 samples and a maximum of 10 samples. Bacterial endotoxin testing is determined on how the product is used. If your product is blood contacting, then it will need to meet the criteria for blood contacting devices. If it does have cerebral spinal fluid contact or any type of brain matter contact, then you would have to meet the requirements for CSF. Gas residual analysis is also performed after both half and full cycles are complete. We want to assess the residual nature of your device after all exposure phases of the sterilization processes. When it comes to performing residual testing, 3 devices is adequate. We like to pull those results into one result so you get an averaged result. We evaluate all 3 EO, ethylene chlorohydrin, and ethylene glycol residuals. Only the ethylene oxide and the ethylene chlorohydrin have requirements to be within a certain level or limit of residue. Your product will either be tested as a limited use device, which is a device that has anything less or equal to 24 hours of exposure to a patient. Or it will be tested as a prolonged use or a permanently used device. Prolonged is anything greater than 24 hours but less than 30 days, and permanent use devices are anything greater than 30 days. And there are respective acceptance criteria for those residual levels depending upon how the product is categorized. So now we go into the acceptance criteria of a clinical batch or a single lot release. When it comes to the acceptance criteria, you'll want to ensure that any data recorded from the sterilization process in regard to the actual chamber itself it does meet the specifications outlined. No growth of the test organism from within the inoculated product or the process challenge devices can be present after the half or the full cycles. For the full cycle process, and this is where the temperature and relative humidity monitoring comes into play, we must ensure the load humidity at the end of conditioning phase is of an adequate amount. Usually anything above 30% is considered adequate, as that helps and aid with the EO interaction in the organism. And we must also ensure that the temperature throughout the EO dwell phase has been achieved or is better when compared to the half cycle. This is where, at the beginning of the clinical batch release, explaining when developing the cycle, you want to make sure that your half cycle parameters are worst case. So when you do perform your full cycle, you'll always have that added measure that your full cycle is going to be at least equal to that of the half cycle process. So for example, if you take into consideration temperature, if you set your temperature at 50 degrees Celsius for your half cycle, then adjusting it to like 52 or 53 degrees C for your full cycle would aid in meeting that criteria. Your bacterial endotoxin of your device must be within the requirements as outlined in ST72. That's depending upon whether it's blood contacting or cerebrospinal fluid contacting. And then of course your EO and ECH residuals of the product will need to be below the allowable limits for how the product is intended to be used. Some additional criteria that you'll need to consider, but usually is not included within the scope or protocol of a batch release, and this is usually done up front, is your stability testing and any type of functionality testing. You also need to take into consideration your packaging. You'll want to, when choosing your packaging, you'll want to identify or confirm, rather, to confirm if it does come with a default shelf life. Some packages do come with that type of claim, but you'll want to make sure that you identify how quickly that the package or that the product will be used after the single lot release. And of course, you'll always want to take into consideration your biocompatibility testing prior to any type of clinical trials. So that outlines the method of performing a single lot release with EO sterilization. Now we'll go into the Annex E and the revision updates. For the purpose of showing you the updates, I am only going to go over the new Appendix E on the right-hand side of the slide. I did just provide the old appendix details for comparison, but we will just go over what has been revised. So the general section was updated to add the clause which states that the single lot release method is not to be used as a mean for routine sterilization. The general section was also updated to outline that the single lot release could be incorporated into a validation protocol or into a validation effort. Section E.2.3 was revised to adjust or reflect the following. Fractional cycles was changed to reflect the wording half cycle rather than fractional, being that you use a half and a full cycle for your single lot release. It was more appropriate to word it as a half cycle rather than fractional. The SAL will be demonstrated by achieving all kill of the inoculated product and PCD following both half and full cycle processes. Prior, it outlined a specification of an assurance sterility level, which is still going to be met and determined based upon the kill of the process challenge device and inoculated product, but it provided some inconsistency in regard to how to calculate that. BI requirements were now outlined and probe requirements were outlined to be follow. The requirements for probes and biological indicators were outlined to follow the tables in Annex C. Prior, there was no reference to that, but you will need to comply either with the Annex C recommendations for probe and BI quantities, or you will need to rationalize those based upon load volume. Then the acceptance criteria was changed to include additional acceptance that wasn't outlined in the previous acceptance. The addition of the criteria to confirm that the half and full cycle process specifications were met when considering the sterilization cycle. And then, of course, there was the humidity and temperature acceptance for humidity following the conditioning phase of the sterilization process, and then, of course, the temperature throughout the EO dwell time. And then Section 7 should be addressed to rationalize and document your product definition and testing. That concludes the summary of the changes in Annex E, and I now turn the time over to Heather, who is going to take the webinar from there.
Thank you so much. Yeah, thank you, gentlemen. We're going to turn over to the Q&A portion of our event. So if you have any questions, you are free to use the widget, the Q&A widget below. Like I said before, if we don't get to all the questions, our speakers are going to follow up with you. So the first question we have is, when is the revised standard being released?
Great question. So in the meetings in March, the committee communicated that the standard is currently in the FDIS process, which is the final draft international standard. This is just the editorial phase where there's no more content change. Again, just editorial and typographical grammar and such. The plan for release is Q2 of 2018.
Oh, did we lose you?
No, I'm still here. Can you hear me?
There you are.
Can you hear me?
Now I can hear you. Yeah, go ahead. You were about to tell us.
Get the answer?
Q2.
Oh, I'm sorry.
Q2 of 20 ...
Q2 of 2018. Sorry about that.
That's all right.
So sometime this
Okay.
quarter.
Is there a phase-in period to comply with the new revision?
Yeah, so right now the phase-in period, there's not expected to be a phase-in period. These changes, and based on the single lot batch release being pretty much a verified process, and the nature of the changes being able to be complied with basically as you start a new process or test plan for a single lot batch release can be made at those times. So again, there's not expected to be a phase-in period. The industry expectation is to comply immediately. Nelson Labs has updated our procedures to incorporate these changes already into our practice.
Gotcha. And with single lot release, does packaging validation have to be completed?
That is up to the discretion of the client. We recommend that they confirm with the packaging manufacturer if it does come with a default time of when the product can be, within a certain time that the product can be used and still be claimed sterile. So a lot of clients do not perform they don't perform any type of packaging testing. At a minimum, I would probably recommend doing some shake, rattle, and roll tests since it will be shipped. But a lot of clients default to using any type of standard claim that the packaging may come with.
Got it. Here's a question. If I have low bioburden recovery when using the inoculated method, what are my other options for retesting to get a higher recovery? Should the recovery method just use an inoculated medium with a higher level of surfactant? If so, how do you quantify that medium?
Sure. So when it comes to bioburden enumeration and validating the method in which that the bioburden is being extracted from the device, you must take into consideration, obviously, the recovery efficiency. Sometimes, depending upon the substrates or materials of the device, may pose may cause an issue in having a really good recovery efficiency. So if that is the case, then you want to not only look into performing the inoculated recovery, but you also want to look into performing different methods of extraction. So if you're just doing manual shaking, for example, to extract the organism, maybe you need to add some additional mechanical agitation in order to get the organism off the device. There's also a method called stomaching. You could look into using the method of stomaching. That's usually the last resort if the manual shaking and the mechanical shaking does not yield a valid recovery. And for recovery efficiencies, anything above 50% is acceptable for medical devices. So as long as you're exhausting your efforts in terms of trying to achieve a recovery efficiency that is sufficient, that is what is expected. Sometimes you're not going to be able to meet that 50% requirement, and as long as you can show that you've exhausted those efforts in how to extract the bioburden from the device, you can use that worst case extraction efficiency.
What's a typical half-cycle EO exposure duration for inoculated product method?
Yeah, so a typical half cycle, if you're using an industrial sterilizer, is anything between 2 to 4 hours is usually a half cycle.
Okay. Can units left over from lot release as part of the full validation still be sold on the market?
So let me just reread that question. Can units left over from lot release as part of a full validation still be sold to the market? As long as you are performing a clinical batch release as part of that validation effort,
sorry about
then
that.
You can use, that's okay. As long as you're performing a clinical batch release as part of the validation effort, then you can use that clinical batch release that's embedded into that effort as release for that product and use it and market it.
Okay. Sorry about that. I've got a new dog, everybody. We're still working on her. Do you think that FDA gives more scrutiny to batch releases?
So no, the FDA doesn't. Right now, one of the main changes that came out, or at least the major push, was to get people away from using single lot batch release for production. Even though the science is fully sound, the expectation and the thought around single lot batch releases, it's not to be used for the purpose of production. So the FDA scrutiny is really more around, are you continuing to use it after you're into production, and you, technically speaking, should have enough data from your single lot batch releases to produce or to conform to a fully validated process. So again, the FDA doesn't have a big problem with the science or the process around it. It's just a matter of what we consider or look at as abusing the allowance for single lot batch.
Okay. We have time for one more question, and this one is there a time frame for completing batch cycles to combine them into a validated cycle? For small manufacturers, the process could take years.
Yeah, that is true. So when considering validation, you're going to take into consideration some cycle development. And when you're performing batch releases as part of a validated process, a validation effort, you're going to potentially have changes over time, more so in the bioburden levels. So when it comes to a validation, usually the time frame of completing a validation is within we say within 90 days. Try to get it completed within 90 days. If you're not going to have clinical units and it's going to be spread over year to year and a half, then what I recommend doing is developing a protocol that outlines the clinical batch, what you're going to do for that clinical batch, but also outlines that the first half and full cycles that you perform for that batch is going to be included as a batch release effort. Once you've accumulated 3 separate protocols, 3 separate batch release efforts, that third protocol you would want to outline it as a validation, and that's where you would summarize everything that you've done over time. The only thing that would be critical in confirming that everything holds water is that you would want to ensure that as the product has been built over time, that everything remains consistent, that the manufacturing location, process, materials, vendors, all those things are consistent over time. Otherwise, it's going to throw into question some of the microbial performance qualification that you did or performed to develop a process challenge device. So as long as you can confirm that the consistency of the product history, then you should be able to apply the validation effort over a significant amount of time. I would say that you would want to try to get it done within a year, though.
That's great advice. Thank you so much. That is all the time we have for this presentation. Again, I want to thank our speakers, David Gilbert and Paul Litley. And thank you to our attendees, and thank you, Nelson Labs. Have a great day, everyone.